Welcome to the webinar on Bayesian Methods for Clinical Studies. My name is Emmanuel Lesaffre, and in about 1 hour I will introduce you to the Bayesian approach. In my regular Bayesian course, I ask the participant what they see. The first reaction is that they see a dog. The second reaction is that they see a black dog. When I ask whether they all agree, and if so, then I say that they can all leave because they are Bayesians. In fact, they use prior knowledge that they've never seen a dog which is black on one side and has another color on the other side. But then I show them this photo of a cat which has two different colors on the head to indicate that prior information can be very useful, but you have to be careful. In our natural life, we are Bayesians. For instance, we don't consult the car's manual every time we drive our car. We combine past experience to know how to drive in traffic. And that's what the Bayesian approach does. It mimics our natural life where learning is done by combining past and present experience. We also are Bayesians in our research. Suppose a small-sized study with an unexpectedly positive result about a new medication to treat patients with oral cancer was presented. The first reaction of the company was probably, that's a great result. But then the clinical experts and investigators, they meet and they look at the medications, similar medications of the past, and they conclude that none of them had such a large effect. So why should this have this large effect? So the second reaction will be, let us be cautious. If that is the case, they're Bayesians. Part of the material comes from a book published in 2012 together with Andrew Lawson and an edited book published in 2020 together with Gianluca Baio and Bruno Boulanger, giving a review of the usefulness of the Bayesian approach in pharmaceutical research. Let us start with the crash course in Bayesian methodology. We look at what is the Bayesian approach, what are the differences with the classical statistical approach, and we use some simple illustrations. The central idea is to combine data where we have assumed a statistical model, so it gives you information on the likelihood, with prior information, with your prior information called prior probability. And that is then combined to update the information on the parameter to result in a revised information for that parameter called the posterior probability. It starts with an elementary property in probability theory saying that the probability that the joint event happens is the probability the first event happens times the probability the second event happens given that the first has happened. This split up can be also done in a different manner. From that result, you receive that the probability that B given A can be written as the probability of A given B times which we call marginal probabilities. This can be further written in a more complicated manner again as conditional times marginal probabilities plus conditional times a marginal probability. Well-known example of Bayes' theorem is the expression of predictive value in terms of sensitivity, specificity and the prevalence of the disease, where then A represents a diagnostic test, a positive diagnostic test, and B represents the event that one is diseased. So let us look at this expression. The sensitivity is the conditional probability that an individual has a positive diagnostic test given that that individual is diseased. On the other hand, the specificity is the probability that one shows a negative diagnostic test given that one is healthy. But the patient is interested in these inverse probabilities. The probability that one is diseased given that a positive diagnostic test was obtained, or that one is healthy given that a negative diagnostic test is obtained. The marginal probability that B happens is just called the prevalence. Now, Bayes' theorem, as we will see on the next slide, allows us to compute the positive predictive value and negative predictive value from sensitivity, specificity, and the prevalence. This is the well-known formula. As an example, let's look at the computerized analysis of brush biopsies to detect dysplasia or carcinoma in patients with oral mucosal lesions, which is the disease, and the diagnostic test is the oral CDX test. This can be found in our book on dental statistical methods, published in 2009. The diagnostic table can be found here, and from that diagnostic table we can read off the specificity in a vertical manner, the sensitivity in a vertical manner, and also we can obtain the prevalence, which is just 26 divided by 96. The positive predictive value can be read off horizontally, and the negative predictive value can also be read off horizontally. We don't need Bayes' theorem when we stick to the prevalence of this example, but once the prevalence changes, we need Bayes' theorem, and then we can use the obtained sensitivity and specificity from that table, combine it with different values of the prevalence. Let us reflect on this example. Suppose that a patient enters the cabinet of a medical doctor and he or she asks, what is the probability that I have that disease? Then the medical doctor, knowing the patient's age and gender, will look at the prevalence of the disease in the relevant population. We could say that it's prior information prior to having the results of the diagnostic test. Then the patient shows the result of the diagnostic test, and then combined with the prior information, the medical doctor will then combine that information using the formula that we just saw, and he gets an updated probability that a patient has the disease or not. This is called the positive or the negative predictive value. Looking back at the formula, we can see if we call this the likelihood, that is actually the information coming only from the data, times the prior, we can see that the posterior is a product of the likelihood times the prior. We need, when we work with statistical models, a more general expression of Bayes' theorem, and this is the basis for the increasingly popular Bayesian approach. Suppose we compare two treatments which is expressed by an unknown parameter theta. For instance, when we work with binary outcome, we can think of odds ratio, relative risk, or absolute risk reduction. When we work with a continuous outcome, we can think of a difference in means. Now, for a Bayesian analysis, we will need to combine prior information on theta with information obtained from current data on theta, then to obtain a posterior idea on theta. What are the advantages of our approach? Well, we can formally incorporate external information. We will see that the output of our statistical analysis is formulated in a much more elegant manner than in a p-value, and in contrast to the computation of a p-value, we don't need to imagine what could have happened but never happened. Finally, the Bayesian software allows us to fit much more complex models than the frequentist, the classical approach, allows us to do. And this is what I wish to show, and to get an idea, I will tell you a story. The story is about the relationship between infant sleeping position and sits. And for a long time the question was, should the baby put to sleep in the front or the back position to avoid sudden infant death syndrome? Here we see a meta-analysis published in 2005 on different studies evaluating whether the baby should be put on the front also called prone or on the back position. And already in 96, carpenters found that the baby should be better put in the back position, but it was not statistically significant. A highly statistical significant result was obtained by Frogat in 1970 and also with Beale in 86. But Tonkin didn't see any advantage of putting the baby in the back position. Here we see the cumulative meta-analysis always combining the result of the current study with the previous study. So we see in 86 a clear evidence that putting the baby in the prone position is not good. But in 1986, Tonkin actually reduced that evidence because in his study he didn't find anything. The conclusion from the cumulative meta-analysis was in fact that already from 1970 one should have given the advice to put the baby on the back. This could have avoided a lot of deaths in infants all over the world. Now here goes the story. Suppose I'm Tonkin and I'm preparing my paper on SIDS in 85. I do a classical analysis on observed data, which are this, and I use a standard statistical program and I obtain for the odds ratio, which is our unknown parameter theta, 0.88 with a 95% confidence interval that includes 1 and with a highly non-significant p-value. Suppose later on I picked up the result of the cumulative meta-analysis after the first 3 studies, and when I looked at the figure, go back here, I looked that this estimate, the best estimate of the odds ratio is around 4.2, and then with this uncertainty. I guess this uncertainty is going from 1.9 to 9.3, so my 95% confidence interval is going from 1.9 to 9.3. What can I do now with this prior information? I can ignore it, but also I can combine the prior information with the current data. But instead of combining all information from the previous 3 studies, I could discount that information. Now, I already have done the case 1, but how to do cases 2 and 3? I didn't use or have to use a Bayesian approach. This is a summary of what we know, or what I know as Tonkin in 86. And here you can see 3 Bayesian analyses, each of them providing an estimate for the odds ratio for SIDS in the front position. You see 3 columns. The first column is when I don't take into account prior information. The second column is when I take the information of all 3 previous studies into account. And the third column is when I take into account the reduced information from the 3 previous studies. You see 3 rows. So in the first row, my prior for the odds ratio goes from basically 0 to basically infinity. So it's telling us that odds ratio can vary or is in between 0 and infinity, which basically says I don't know it. In the second case, I take the estimate from the cumulative meta-analysis with a 95% confidence interval, and in the third case, I enlarge that confidence interval. These are the estimates from the data. They always are the same because the data don't change. Then I combine prior information with data information and I get this posterior. This first posterior, when I actually didn't make use of any prior information, that posterior information is basically the same as when I look only at the data. When I combine the cumulative meta-analysis information with the data, my estimated odds ratio is pulled towards 4.2. With a 95% Bayesian confidence interval now, excluding 1, so indicating that putting the baby in the front position is bad. When I use the reduced prior information, I also see that the odds ratio is moved towards 4.2, but it's actually, compared to the previous one, pulled towards 1 more, with a larger Bayesian confidence interval, now excluding 1. What are the findings of our Bayesian analysis? When I didn't assume anything, then the result of my Tonkin study is just simply obtained. When I used the results of the 3 previous studies, then, as you can check in the cumulative meta-analysis, I actually reproduce the result after 4 studies. So this confidence interval and result is actually the result that you can find somewhere here on the 4th line. But when prior information on the risk is reduced, then we see that the odds ratio compared to the previous one is pulled towards 1, but is still larger than 1. Let us now see how to specify the prior information on theta. This is the result of the first 3 studies in the meta-analysis. And we graphically show it here with the estimated 4.2 for the odds ratio and the 95% confidence interval. So any prior information, any prior density should reflect this. Now we have to realize that an odds ratio is a continuous variable going from 0 to infinity. So it doesn't make sense to put probability on a particular value because it's always 0. So what we do is we give it a density that roughly represents the information that we obtained from the 3 previous studies. So we see here that the maximum value of this density is around 4.2, and that the area under the curve under that density is 0.95, corresponding to the 95% confidence interval. So you could say that this density expresses our prior knowledge of the odds ratio coming from the first 3 studies, where intervals have an interpretation of probability, but where values or probabilities on values don't have an interpretation. We don't have to specify the probability on the odds ratio, we can also specify it on the log odds ratio. We can see here that the probability, the prior density on the log odds ratio looks to be like a Gaussian distribution. Now, specifying a symmetrical prior density on a parameter is useful, not only in classical statistics, but also in Bayesian statistics. And you can see immediately why. If you want to specify a prior distribution that basically doesn't represent any information on the parameter, then it's easier to do that on a symmetrical scale, on a symmetrical density. Here we see a vague prior on the log odds ratio, because we see that the log odds ratio goes from minus 20 to 20, which basically says on the odds ratio scale, a value close to 0 and a very high value. This is what we know that the odds ratio should be lying in between. This is called a vague prior. It's actually sometimes also called a non-informative prior. You could say that this prior is in fact a bit too vague because it puts some prior probabilities on unrealistic odds ratios. Now, what information do the data bring on theta? Well, this comes from the results, my results in '86, and this can be represented by a likelihood function. It comes from a particular statistical model that we assumed for our 2x2 table, and then we derive the likelihood function, which is the probability or the plausibility of each parameter theta that we can assume, in this case each possible odds ratio, given the data. So now we have 2 sources of information on theta, the prior and the likelihood. So what we do is We combine these by taking just simply the product of these two functions for each possible value of theta. And we standardize it, because we want, at the end of the day, again, a density. And that's Bayes' theorem. Graphically, we get the following. So we have the three cases: vague prior, the prior from the cumulative meta-analysis, and the discounted prior. Let's look at the first case. The prior is very flat on the range that we are looking. So from minus, let's say, minus 1.5 to 3, this Gaussian distribution looks basically horizontal. Combining it with the likelihood, we get a posterior that's basically the same as the likelihood, which basically says we haven't learned anything. This is the informative prior coming from the 3 previous studies, combined with the same likelihood, and we get this posterior. This posterior is a kind of compromise between what the likelihood says and the prior, and is also more peaked, which indicates that we have learned. When we discount the prior, so you can compare this prior to that prior, then you can see that this prior has a higher dispersion. So it means there is less information in this prior, and you can see that also in the posterior, which is still a compromise between likelihood and prior, but is less peaked. Now we can express our posterior, of course, also on the odds ratio. So here you see the posterior again on the log odds ratio, where you can determine the 95% posterior probability so in other words, we can determine the interval where that corresponds with 95% posterior probability. Important to mention is that in the Bayesian context, this contains the most plausible values of the log odds ratio, here the most plausible values for the odds ratio, in contrast to what the 95% confidence interval in the classical approach tells us. In the classical approach, that interval says that when we repeat our study, in 95% of the times the true value is inside that 95% confidence interval. But it doesn't tell us anything what we believe in that of that interval, that it should contain so much that it contains the most plausible values. With such a posterior probability, with such a posterior density, we get all kind of derived measures, like the posterior probability that the odds ratio is bigger than 1, which relates to the p-value, but is easier to understand. But we can also get the most probable value for the odds ratio when we read of the value where that posterior density is maximal, the posterior mean, median, and standard deviation. And as I already said, the Bayesian 95% confidence interval. After showing this analysis or this story, an often posed question was that the participants asked whether the Bayesian analysis is just a cumulative meta-analysis. And the answer is, of course, no. The Bayesian approach offers us much, much more, because the prior knowledge can come from everywhere, not just from in the context of a meta-analysis, and can be applied to basically every problem. Also, the Bayesian output, as already shown, is much more useful than the p-value. Finally, the Bayesian software can handle problems that the classical approach cannot. This leads us to the formal definition of Bayes' theorem, where we work with an unknown parameter theta and data, where we have a likelihood that expresses the plausibility of theta given the data for a grid of theta values, and where we have prior information on our theta parameter. Very important to mention is now that in the Bayesian approach, the unknown parameter is a random variable, which is not the case in the classical statistical approach. Combining now the likelihood times with the prior, we get the posterior, and it's just the product, likelihood times prior, divided by what we say constant. So we read off the posterior density of theta after having seen the data is a combination of the likelihood for theta after having seen the data with the prior on theta, divided by a constant. And what is that constant? Well, it's just the area under the curve of this function. And that brings us to this integral. So the posterior is a standardized product of the likelihood and the prior, and the posterior and the prior attach Bayesian probabilities to the different theta values, basically intervals of theta values. And so the unknown parameter gets a distribution quite different from classical statistics. Note that in the classical approach, one often makes use of this Bayesian notion when, for instance, we work with the 95% confidence interval. As I just mentioned, this is usually interpreted as the interval that contains, with 95% probability, the most plausible values of theta, even in the classical approach. Now, in our daily work as a statistician, we usually work with more complex models. Take already the normal distribution, which has a mean and a variance, so two parameters. Or linear regression, where we have regression coefficients and residual variance. Logistic regression, for instance, where we have regression coefficients, and so on. Now, Bayes' theorem is exactly the same for a single parameter as for multi-parameters. So you can see it here. Only difference is that theta is now a vector. So we get the same expression as before, but now for a vector. But what's more complicated is that the prior must be specified in a multivariate manner. In practice, we see that often that prior is taken as the product of priors on each parameter separately. That makes our life already a bit simpler. But posterior is still multivariate, and that makes our life complicated because it means that this integral has the dimension of the parameters. So if we have a 10-dimensional parameter, this is a 10-dimensional integral, which is hard to compute. But then we can't work with this multivariate posterior. So we have to work with posterior densities of each parameter separately, which again involve integrals. And these integrals can be very complicated. So we see that to apply Bayes' theory, one needs to compute an often intractable integral. And it took about 250 years before one found a solution. And the solution was in bypassing the integral by sampling the posterior and thereby approximating also the posterior summary measures. And that sampling algorithm, or that solution, is called Markov chain Monte Carlo sampling. And together with the software Winbugs, it caused a revolution in Bayesian statistics, but I could say in general in statistics. After the introduction of Winbugs, also other software appeared, like OpenBugs, JAX, Stan, Inla, plus links to R in Python, and many dedicated R and Python Bayesian packages appeared since then. We could say that instead of adapting the statistical model to the software, which often happened in my practice in the past, the Bayesian software allows us to swiftly adapt the program to the statistical model. Here you can see a bugs program for the infant sleeping position relating to SIDS. We needed MCMC computations for this example. And here you see the Winbugs program. So you can see a kind of R statements where we say that here the number of events have a binomial distribution for the front position and the back position. Then we compute here the log odds ratio, giving it a prior, and we compute here the odds ratio, and so on. We typically see trace plots to see whether our computations go in a good direction, and if we see a stationary plot, which we call trace plot also often a time series plot, if we see a stationary plot, we conclude that our sampling from the posterior goes well. And if it goes well, we can compute the marginal probabilities of each of our parameters. This is the marginal posterior distribution of our odds ratio. And this is the marginal posterior distribution of the probability of events in the front position. We can also get posterior summary measures. For instance, this here is the posterior mean of the odds ratio with its posterior standard deviation, indicating the uncertainty, and then with some correlation or Monte Carlo estimates of our estimated posterior summary measure. This is for the log of the odds ratio and so on. All of that can now be applied to pharmaceutical research because we can often have some idea of the efficacy of a treatment versus control. In other cases, when we can be skeptical that the new treatment is better than the control. And in most of the cases, maybe we will not have any idea of the efficacy of the new treatment. In all these three cases, we combine that with the results of our clinical trial. And how to do that formally, well, that's again using Bayes' theory. Now, why a Bayesian approach is useful or even maybe necessary in the design and analysis of clinical trials will be seen after the commercial spot we see immediately. And in session 2, we will look at the usefulness and the integration of the Bayesian approach in various research areas. The Bayesian approach to inference is increasingly popular in various application areas, like pharmaceutical research, genetics, epidemiology, psychology, and so on. Let us look at pharmaceutical research. Well, in our book in 2020, we gave an overview of the use of the Bayesian approach in product development and manufacturing, process development and validation, analytical methods and assays, and blah, 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 and so on, medical devices, to indicate what could be the usefulness of the Bayesian approach in pharmaceutical research and to promote it. This is a byproduct of the annual meeting on Bayesian Methods we call Bayesian 20XX. For instance, this year is the Bayesian 2024 meeting organized, where we meet or academic statisticians meet with statisticians from the pharmaceutical research, pharmaceutical industry. This is a popular program, Bayesian program in genetics. Here you can see 3 books from authors who are working in Bayesian methods, working in Bayesian methods in epidemiology, in forensics, in archaeology, where you usually have a lot of prior information but not much data. In ecology, very important application area to look at the geographical spread of certain species and also the evolution of that spread over time. Even in theology it is used. And many other applications areas you can find where the Bayesian approach is used. Let us look now at applications of the Bayesian approach in clinical trials. So here we will illustrate in this session various applications of the Bayesian approach in clinical trials. Of course, the traditional approach to statistics can still and will still be used. Definitely when there is enough budget. It means that if there is enough budget to recruit a lot of patients, and if the recruitment of patients goes fast, there's usually no difficulty. And when the control group can easily be taken, and when it's ethical to set up a clinical trial applied to children, pregnant women, and so on. There is no problem then. But what if that is not the case? The frequentist approach is still the most used approach in the setup, conduct, and analysis of clinical trials. And this will still be the case for a long time, but we can see something changing. For instance, in the phase 1 trials, dose finding and safety trials, the Bayesian approach is one of the important tools nowadays. In phase 2 trials, proof of concept trials, the Bayesian approach is the preferred method in some of the major pharmaceutical companies, and in medical device trials, the Bayesian approach is one of the standard tools. But in phase 3 trials, the Bayesian approach is only used in interim analysis, but maybe things are different in the near future. Here's an illustration of some simple Bayesian analysis of a randomized controlled trial, just to fix the ideas. And the first is a study that was done some years ago, the Aristotle study. It's a large randomized controlled trial, originally analyzed in a frequentist way, and it is a comparison of apixaban versus warfarin in patients with atrial fibrillation. Apixaban and warfarin is given to patients with atrial fibrillation to avoid the formation of clots. They are called novel oral coagulants. Actually, apixaban is called, warfarin is the standard. The second study is the well-known Pfizer study to show the effect and safety of the vaccine that was used to treat patients with COVID. Let's start with the Aristotle study, which is a double-blind randomized controlled trial to compare apixaban with warfarin, and was based on a large number of patients, namely around 18,000. The primary outcome was ischemic or hemorrhagic stroke or systemic embolism, and the primary aim was to test for non-inferiority of apixaban versus warfarin, and in the second objective was to show superiority. A survival analysis was done, and for the non-inferiority test, the threshold of 1.38 for the hazard ratio was taken. So, to refresh your mind, the non-inferiority test is a test where here apixaban is shown to be, or possibly shown to be, slightly worse than warfarin, while in the superiority test, here is the test to see whether apixaban is better than warfarin. The results were on a median duration of 1.8 years, for apixaban 1.27% per year, and for warfarin 1.60% per year, which shows a hazard ratio of 0.75 in favor of apixaban, with a 95% confidence interval excluding 1. The one-sided p-value for non-inferiority was smaller than 0.01, and the two-sided p-value for superiority was smaller than 0.01. Now, for our Bayesian calculation, and to make life a little bit simpler, we assume that all patients were followed up for 2 years, and that we are interested in the proportion of patients that experience the primary event. So we have pi a, unknown proportion for apixaban, pi w, the unknown proportion for warfarin, and delta, the unknown efficacy for apixaban versus warfarin. The 1.38 non-inferiority threshold for hazard ratio is translated into a difference, a delta non-inferiority of minus 0.01. The original non-inferiority test, which was done at a one-sided 0.05 significance, is translated into a posterior probability that delta is bigger than this difference, that posterior probability is bigger than 0.95. And for the superiority test, which was originally done at a two-sided 0.05 level, we demand that the posterior probability that delta is bigger than 0 should be bigger than 0.975. And we consider 3 cases. The first case, that we don't have prior information, then we use a vague prior. Second, in the second case, we used an informative prior for warfarin, with the reasoning that warfarin is a standard drug, was a standard drug for many years for atrial fibrillation patients. So why should we ignore that information? We found that the primary outcome is around 3% for warfarin patients with warfarin atrial fibrillation patients with 95% uncertainty between 2 and 4%. And the third case, we take a skeptical prior for delta. And that expresses that we doubt that the new medication, apixaban, is actually better than warfarin. And we do that by giving it a prior around 0 and with 95% uncertainty between -2% and 2%. And we considered two study sizes. First, a smaller study size than the original, and then second, the original study size. Here are the observed proportions, and here are the results for the smaller study size and this for the total study size. In the first case, we for the vague non-inferiority test, we get a non-inferiority probability of 0.997, really satisfying non-inferiority. On the other hand, the superiority test gave us a posterior probability of 0.8, which was much lower than what we required, namely 0.975. When we took the informative prior for warfarin results, then the non-inferiority test was basically the same, but the posterior for the superiority test increased with 6%. On the other hand, when we took the skeptical prior for delta, then also the posterior probability for the non-inferiority test decreased, and it decreased much further for the superiority test. When we took the total sample sizes, then we saw basically no effect on the non-inferiority test, and we saw a similar effect for the superiority test. We show this also graphically. This is the prior in the first case. The posterior is basically the same as the likelihood. This is the prior for the proportion on the warfarin in the second case, and then we can see that the posterior is more peaked than the likelihood and definitely more than the prior, and that information seems to be used therefore. The third case is where we put a skeptical prior on delta, and we see that the posterior is a bit more peaked than the likelihood, but we see also that the posterior is pulled towards 0, which is the effect, of course, of that skeptical prior. In the first case, we again conclude that the frequentist result is equal to the Bayesian result. So again, we can show that Bayesian analysis can actually replicate frequentist analysis. This is the Pfizer study showing the safety and efficacy of the vaccine that they used to treat patients with COVID. And on this slide you can see that they used a credible interval, which is another term for Bayesian confidence interval, and they also used the Bayesian beta binomial model, which simply says that they combined a binomial likelihood with a beta prior. So surprisingly, this was a huge study, and still they made use of the Bayesian approach. One of the reasons what you could say is that the Bayesian approach gives us more intuitive results, conclusions that are better to understand than just reporting p-values. The Bayesian approach is also useful when in our clinical trial the data have a non-standard distribution, or when the distribution is relatively complex, or when the data structure is relatively complex. Now that is not necessarily due to the Bayesian approach, but is more due to the flexibility of the Bayesian software, namely the MCMC approach. So we can have, for instance, easily dealing with a situation that the response has a non-standard distribution like the t-distribution or a skew normal distribution, or when there are missing data in the response and covariates, or when some model parameters are constrained. This is no difficulty when you use Bayesian software. And Bayesian software is actually the preferred software when you do joint modeling of longitudinal and survival outcomes and longitudinal models of mixture distributions. Let's look now at the use of Bayesian methods in interim analysis. So we do an interim analysis to verify some safety issues before the study has ended, or we want to do an interim analysis to check whether we can stop for efficacy or stop for futility. We show now an example of the Bayesian approach for interim analysis. We have to realize now that the Bayesian approach does not correct for multiple views. So stopping only depends on what we have as information on the treatment effect, in this case the posterior probability. We can also make conclusions when we base it on the posterior predictive distribution. Now the posterior predictive distribution is the distribution that tries to imagine what will happen when we continue our study until the end. Let us look at the compile study as an example where the Bayesian approach was used for checking at interim the results. And this is a recently finished study in 2021 coordinated by the New York University to evaluate the effect of convalescent plasma on COVID-19 patients. And this is an example of a meta-analytic approach. Meta-analytic because there are many studies combined together, pooling individual patient data from completed at that time or early terminated and ongoing studies. 8 randomized controlled trials to check the effect of convalescent plasma. In this case, the convalescent plasma is always the same, but the control groups differ. So this is a multi-country, multi-center study utilizing continuous monitoring using Bayesian stopping rules. And those Bayesian stopping rules were motivated because the frequently stopping rules, the thresholds to determine, was far too complicated. But also because the use of posterior probability as tool for monitoring is much more intuitive. However, because of regulatory demands and rules, computer simulations were needed to establish the control of the type 1 error rate. The primary endpoints was the WHO clinical status at about 14 days and whether mechanical ventilation was used or was worse. So here's the WH scale, where 0 is when the patient was unaffected, no viral load was detected, and 10 when the patient died. There were 2 primary analyses. They were all done using a Bayesian approach. Outcome 1 was for outcome 1, so the WHO outcome, an ordinal random effects logistic regression analysis was used, and in the second, a binary random effects logistic regression analysis was used, where the random effects was a trial. In all analyses, age, sex, sex, and other covariates were taken in the models. Of course, for a Bayesian analysis, all model parameters have to be given a prior distribution, and most parameters have been given a vague prior, but for the effect of convalescent plasma, a skeptical prior was taken. And Bayesian monitoring was based on 2 posterior probabilities for each outcome. We stop if the odds ratio was smaller, odds ratio being smaller than 1 was bigger than 0.95 in favor of convalescent plasma, and that the same odds ratio was the posterior probability that the same odds ratio was smaller than 0.8 was bigger than 0.5. For the binary outcome, similar demands were necessary. So both outcomes were evaluated, and for each outcome there were 2 requirements to be satisfied. An extensive simulation showed that this was the right approach. A number of interim analyses were done in November 2020 and 2020 and so on. And the final analysis was done and May 21. Unfortunately, the criteria were not met, so it could not be concluded that convalescent plasma was beneficial for all COVID-19 patients. And what you see here are the posterior probabilities for the odds ratio on the, in this case, the cumulative odds model. So that's the first outcome. So this is the posterior probability that we are interested in, and that is for the binary outcome. Yeah, that's for November 2020, January 21. So what we ideally would see is that this posterior probability goes towards the negative side, also towards left. And this was the final posterior probability for the cumulative model and for the logistic model. And again, what we are interested in is this posterior probability and that posterior probability, which was unfortunately not big enough to conclude that convalescent plasma was useful. Another application area of the Bayesian approach is in dose finding studies in phase 1 cancer studies. So we want to find in this study, in this kind of study, the highest dose with an acceptable toxicity. So what we want to find out is the maximum tolerated dose to be applied in phase 2 later on. And the toxicities that are used to define the maximum tolerated dose are the dose-limiting toxicities. Now, the characteristics of phase 1 dose escalation studies is that we usually have late-stage patients in cancer, very small-sized trials, and a mix of tumor types. There are two main approaches. First, the rule-based approach, which is also called the 3+3 rule-based approach, or the model-based approach. And that second one is based on Bayesian modeling. The rule-based approach, which is graphically depicted as follows, is we continue to increase the dose if there is less or equal to 33% patients who suffer from a dose-limiting toxicity. The difficulty with this approach is that the final maximum tolerated dose is based on too few patients. On the other hand, when we apply the model-based approach, that's based on Bayesian modeling, and the two most popular approaches is the continual reassessment method and the escalation is the EWOC method. I forgot about that. They assume a particular monotonic relationship between dose and the probability of dose-limiting toxicity, and they dynamically adapt the dose at each subject based on previous observations. Then you have a dose adaptation based on the predicted toxicity at each possible dose, and the prediction is based on a model for toxicity estimated from previous observations. The advantage compared to the classical design is that the probability of toxicity at a specific dose is based on all observations at all doses. An important application area of Bayesian approaches is for orphan diseases. A rare disease, also called orphan disease, is any disease that affects a small percentage of the population. And recently there is an inflation of such rare diseases due to subpartitions of the diseases. A major difficulty with rare diseases is that huge sample sizes are needed, and use of prior information is then an increasingly popular way to reduce the sample size. And here's an example of a drug that was approved by a regulatory agency after a Bayesian analysis. This is the hemophilia safety study. Hemophilia A is a genetic disease in which the clotting factor 8 is deficient with an increased bleeding tendency, and that can be life-threatening. Both are rare diseases. The treatment of the disease can either using donated blood plasma or DNA recombinant factor 8. But both have adverse events. The first blood donation can lead to transmission of diseases. The second can lead to the development of inhibitors. Now in 2005, Refactor AF was developed, which is a particular procedure to produce DNA recombinant factor 8. It was developed and one had to show its safety. At that time, it was known that in 4 other similar compounds, 6 out of 328 patients developed an inhibitor. Now, the FDA stated that for the trial to be successful, the upper bound of the two-sided frequentist 95% confidence interval for the product inhibitor incidence must be below 6.8%. Sample size calculations revealed that the requirement of FDA was unrealistic. They needed about between 7,000 and 50,000. So the company suggested a Bayesian scenario, and that was accepted by FDA. On this page you can see that several Bayesian reasonings were applied to actually come up with a new rule. That new rule suggested that they used, instead of the 6.8%, they used 4.4%. Then based on previous data within the company, they combined prior information coming from these previous data with the current study, and they study results based on 90 patients, and they obtained that the upper bound of the 90%, 95% equal tail credibility interval was below that new threshold. We finished this webinar by discussing very briefly some additional but very important application areas of the Bayesian approach. The first is when you have historical information that is useful for the analysis of the current study. So often it occurs that within a research institute an experiment has been repeated several times with the same control group. How can we use that information? So this brings us to the question, how much prior information can and should be used? And this is the topic of Bayesian borrowing information approaches. So this actually, when you go back to the hemophilia example, this was an example of information borrowed from the past. Two big approaches are the power prior approach, which was applied in our case on Hoven studies. This is a kind of studies that were done in the Erasmus Medical Center where I worked, and the meta-analytic approach. There are also other approaches like the commensurate prior and so on. So this is really a rich area of research, and the application areas are in orphan diseases, pediatric studies, bridging studies. Medical devices is an important application area for Bayesian methods because it's quite a different area. It's different from drug research, and here you can see a couple of reasons why this is different. It's also here where the Bayesian approach is really accepted by FDA and EMEA. Finally, Bayesian adaptive trials. Adaptive trials is a design that is based on the results of the trials so far. Two-stage designs are an example, dose-finding studies, adaptive randomization, seemingly phase 2 or 3 trials, master protocols trials, platform trials, umbrella trials, basket trials. They all have the aim to speed up the pharmaceutical process. And the Bayesian approach plays an important role in this. Here you see quickly what this can involve. It can involve adapting doses, removing arms, and so on. Here is more information, what is a platform trial, and here what you can see is what is a basket trial, an umbrella trial. All of these new developments are major application areas of the Bayesian approach. So, very nice, very useful, the Bayesian approach. There are many additional topics I could have talked about, but still there are obstacles for the Bayesian approach. In the past it was that there are no computational tools to do a Bayesian analysis in practice, and now is tradition, the tradition of doing a frequentist statistical analysis, but also that the master programs in statistics have often little training in Bayesian methods, the religious status of the p-value, and maybe other things. But good things are coming maybe, is more willingness to accept a Bayesian application. Thank you for your attention.